So where were we? We were in the Bohr effect, weren't we?
Yeah.
But I went ahead and added this animation just to show you what how oxygen binding just affects the overall, what's this called? Our conformational change from the, what state?
T state to R state?
Yeah, the tense state to the relaxed state. You can see the tense state here and then the relaxed state once it's binding. So I thought that's a cool little GIF animation. So I just threw that in there because this is the last figure we did, showing you the difference, up close at the actual F helix where you get the binding, where that this histidine coordinates the iron and the heme, and you get that structure. So, but now we're going to talk about the Bohr effect. Anybody know what the Bohr effect is?
Effect on the pH.
Yeah, so it's the effect of the affinity on the pH, basically. So why would that be important for transporting oxygen to the periphery?
Because the pH has like an increase.
The pH doesn't increase in the periphery.
Well, it has to do with like the H plus.
Yeah, the H plus concentration increases, which causes what to happen to the pH? Yeah. So you have more hydrogen ions due to metabolism. So the pH is lowered in the periphery versus where the lungs are, or versus the lungs. And then the hemoglobin affinity for oxygen depends on the pH. So do you think it's going to have a higher, well, it probably says up there. It's going to have a higher, does it make sense that it would have a higher or lower affinity at a lower pH? At a lower pH? No.
Oh wait, for oxygen?
Yeah, for oxygen.
Okay. Oh yeah, no, lower for higher for hydrogen then.
Yeah, so because in the periphery where you have all the metabolism going on, the pH is lower. That's where you need the oxygen to be dropped off. So if you have a lower affinity, if the hemoglobin has a lower affinity for oxygen, it's more likely to get rid of more oxygen there where the pH is lower, where oxygen is needed for metabolism. Does that make sense? So the pH difference between the lungs and the metabolic tissues increases with efficiency of oxygen transport, and this is known as the Bohr effect, is this pH effect on the affinity for oxygen. And how it works basically is, so you got protonatable groups on the hemoglobins, and one of them is histidine. Why is histidine like a protonatable group? Do you guys remember the pKa of histidine side chain? It's got that imidazole ring group. So the pKa is, I think, around 6.5, so it's right around 7. So it can exist either protonated physiological pHs or unprotonated. So this is where you get the protons added in the hemoglobin, and it actually forms a salt bridge between a nearby aspartic acid. So that salt bridge is going to help tense up that hemoglobin, right? Make it in the T state, stabilizes the hemoglobin. So the T state, is that higher affinity for oxygen or lower affinity for oxygen?
T state's got lower affinity for oxygen.
Right, yeah, so the tense state. Exactly. So that's what happens with increased protons in solution. It's going to protonate the histidine, causing a salt bridge, and then you're going to have an increased tense, T state of the hemoglobin. And this is what the saturation curves look like for hemoglobin at different pHs. Can you guys understand how these work? Read them? So as you move along the bottom of this axis, you're increasing the concentration of oxygen, the partial pressure of oxygen. And this is the fraction bound, right? So if you look at the 50% bound, you need less oxygen for 50% bound at pH 7.6 than you would for 7.4. You need a little bit more concentration of oxygen than you do for 7.2. And that's what happens in the lungs. You're probably around 7.6, and as you go to the periphery, it's going to work its way to 7.4. And rather than just, let's say the partial pressure oxygen's at 2, rather than, if you go from, let's say 8 to 2 in the periphery, rather than dumping off this much, you'd get you would actually go all the way down to like a quarter, or 0.25%, or a quarter, 25% oxygen bound, based just on pH changes. Carbon dioxide also is a product of metabolism, right? That you're going to have increased concentrations of carbon dioxide, and hemoglobin actually will transport carbon dioxide itself. So carbon dioxide is produced by metabolism in the tissues, and it has to be exported. 15% of this, 15 to 20% of the carbon dioxide is exported in the form of carbamate ions. And what happens is the carbon dioxide reacts with the amino terminus on the hemoglobin and actually forms a carbamate ion on each of the polypeptide subunits. And this actually yields another proton, right? So when you form that carbamate ion, you're actually lowering the pH even further than you would with just metabolism, which contributes to the Bohr effect even more. And then the carbamate forms additional salt bridges that are stabilizing that tense state itself.
Do the two forms always form salt bridges, or is it sometimes hydrophobicity that tenses out?
It's going to be the salt bridge that tenses out due to this effect.
Okay.
So, and then the rest of the carbon dioxide is exported as dissolved bicarb and formed carbonic anhydrase, and also produces a proton when you form carbonic acid, and then that is going to dissociate to form bicarb and another proton, right? But this shows you just the overall reaction. So you have the N-terminus of the hemoglobin reacts with the carbon dioxide. You form carbamate plus a proton. So I don't think I had these. I just changed these today, so sorry. Another regulator of affinity is this compound 2,3-bisphosphoglycerate, which regulates how oxygen binds. It's a negative heterotrophic regulator of hemoglobin function. So do you guys remember what the heterotrophic and homotrophic regulators of, are? What we're talking about? Remember, homotrophic is where the regulator of affinity is actually the substrate, the normal substrate. Heterotrophic is a different binding site and a different substrate, and that's what 2,3-bisphosphoglycerate does. It's present at micromolar concentrations in your erythrocytes, so erythrocytes produce it, and it's produced from an intermediate in glycolysis. And it's a small negatively charged molecule. You notice there's 1, 2, 3, 4, 5 negative charges on it, so it's a lot of negative charge. And what it does is it binds to the positively charged central cavity in hemoglobin. And again, it's like you're forming more salt bridges, sort of, not between the protein, but between the protein and the substrate. And it stabilizes this T state even further. And this is a picture of 2,3-bisphosphoglycerate binding in the central cavity of hemoglobin. You've got 4 subunits, alpha 2, beta 1, alpha 1, and beta 2 all together, and it binds here. And if you notice, there's histidines, positively charged histidines, and these are probably going to be lysines and such. A lot of positive charge in the central cavity, and then that's where you get the binding of the 2,3-bisphosphoglycerate, which stabilizes, again, that tense, the T state of the hemoglobin. This is the one from your book. T state, the 2,3-bisphosphoglycerate, and the relaxed state right there. So this has the effect, again, so your book says that, my old book would focus on the effects of 2,3-bisphosphoglycerate and fetal hemoglobin. Our book, the new book, focuses on how it's an adaptation to altitude change. So at higher altitudes, we end up with higher concentrations of 2,3-bisphosphoglycerate, which then shifts that. If you have higher concentrations of 2,3-bisphosphoglycerate, it shifts that curve to the right. This is, again, your oxygen saturation curve, which lowers the affinity for oxygen. So you're going to be able to dump off more bound oxygen at the periphery with the 2,3-bisphosphoglycerate there. Also, I don't think you're in the book, Does it talk about fetal hemoglobin at all? In reference to 2,3-bisphosphoglycerate? So fetal hemoglobin, you know, in development fetuses, they go through multiple different stages of the hemoglobin. And near the end, the fetal hemoglobin, it has a lower affinity for the 2,3-bisphosphoglycerate. So there's less 2,3-bisphosphoglycerate bound to the hemoglobin of fetuses. But the mother's hemoglobin has normal affinity and normal amounts of bisphosphoglycerate. So that's how fetal hemoglobin ends up having a higher affinity for oxygen than the mother's hemoglobin, and you can transfer the oxygen across the placenta that way. And then spectroscopic detection of oxygen binding to myoglobin. And the heme group is actually a strong chromophore that absorbs both in the UV and the visible range. And you can tell it absorbs in the visible range, right? Because it's blue, right? When it's deoxygenated, and then it's red. Oxyhemoglobin is very red, right? So the ferrous form, Fe2, without oxygen has an intense Soret band at 429. The oxygen binding alters the electronic properties of the heme and shifts that position to 414. So spectroscopically, you can tell whether it's bound or not, right? So sickle cell anemia, so that's due to a glutamic, actually amino acid number 6 mutation causes, it's a point mutation in the DNA, so just one nucleotide in the DNA changes, which alters what should be a glutamic acid, right, to a valine. Can anybody tell me the differences between glutamic acid and valine as far as amino acids go?
Valine's got 5 binding sites.
5 binding sites? Or what do you mean?
Well, other than the binding sites, I mean, glutamic acid, I don't know about glutamic acid. I just know valine's got 5 binding sites.
So glutamic acid, from the name, you can tell what kind of charge is it going to have?
Negative.
Yeah, so it's negatively charged. Valine, is that a charged amino acid at all? Yeah, it's nonpolar, so that's a hydrophobic amino acid, which is going to want to be buried, you know, in the center of a globular protein rather than be exposed to the solvent, which this part of the hemoglobin is exposed to the solvent. So here you have one valine hydrophobic molecule out exposed to water, and it doesn't like to do that. So what happens with sickle cell, so the valine, it's going to bind to a different hemoglobin molecule to form a strand. So what happens is, because all those valines are there, they'll interact with each other and you'll form polymers. I think your next figure shows that. You form these long polymers of hemoglobin and you get crystallization formation. And this is a cool, I couldn't get it to work on my computer earlier. Oh, I'm going to the wrong thing. I've got to go over here, don't I? I guess I didn't make it a link. Never mind. It's a cool website where they take you through the crystal structure. Like, you cut through it and look at actually like all the different amino acids and how it'll polymerize. So it didn't work on my computer because we don't have the right Flash or Java, I think. Anyways, 2 types of immune cells. Who's taking immunology right now? Most of the class, I think, right? So I've never taken an immunology class, so I guarantee you guys know more than me already. So we've got what? Cellular immune system targets its own cells that have been infected, also clears up virus particles and infecting bacteria. Key players, you have macrophages, killer T cells, and inflammatory T cells, Th1s. Have you guys learned about these? And then the humoral or fluid immune system targets extracellular pathogens, right? And they make soluble antibodies. Do you guys know the structure of an antibody?
Light chain 17.
Yeah. So that's all this section is about, pretty much, showing you how antibodies will bind to fragments displayed on macrophages, or you have an FC region coated of IgG coated virus, so you have a virus gets coated with antibody. FC receptors are coating macrophages. The FC, which is basically the bottom part of the Y, right, on the antibody, will bind to its receptor, causing phagocytosis. The phagocytes ingest and destroy the viruses that way. The humoral immune system, vertebrates also fight infections with soluble antibodies that specifically bind to antigens. So some definitions that you probably already know, right? Antigens are the substances that stimulate the production of antibodies, right? Have you guys heard of haptens? Did he go over haptens and antigens and all of that? So antigens are what the antibodies bind to. Typically macromolecule in nature, recognized as foreign by the immune system. Coat proteins of bacteria and viruses, and carbohydrates of cells or viruses. Antibodies are proteins that are produced by B cells and specifically bind to antigens, and the binding will mark the antigen for destruction or interfere with its function. And a given antibody will bind to a small region, which is another definition, an epitope, right? And one antigen can have several epitopes. And if you have a polyclonal antibody that recognizes one antigen, that polyclonal antibody will recognize several epitopes, right? But a monoclonal should only recognize one epitope. What's the difference between monoclonal and polyclonal?
Antibodies.
Yeah, well, the polyclonals are produced by multiple clones of cells. Monoclonal is just going to be produced by one particular B cell, or in tissue culture, one that's made from one clone, made from one B cell. So it's just one type of antibody that's being produced there. And as you guys were telling me, composed of 2 heavy chains and 2 light chains, IgGs are in different domains. There's a constant domain and a variable domain. The constant domain has a constant structure to it. The variable domain is variable depending, and that's where you get binding to specific antigens or epitopes. Then you have light chains and heavy chains. Both light and heavy chains have one constant and one variable domain, well, except for the heavy chains have 3 constant. But still, there's a constant region and then one variable domain on each. And then the variable domains of each chain make up the antigen binding site. You have 2 antigen binding sites per antibody, and the variable domains contain regions that are hypervariable. Specifically, the antigen binding site confers a high antigen specificity. And this is your overall structure again. So constant chain, and then your light chain, your variable domains, and then your constant regions. And then variable domains again. And they're held together, light chains and heavy chains held together with disulfide bonds. The 2 2 heavy chains are held together by disulfide bonds, and again, the light and the heavier disulfide bonds. Your FC fragment is this portion, so that's what's recognized by the FC receptor, and then your variable fragment is this portion. And you can actually take IgGs and treat them with papain just to purify the FAB fragment, if, for say, you didn't want to cause some inflammatory response and interact with that. So a lot of drugs are made just to FAB fragments. And this is a ribbon diagram of a IgG with the bound carbohydrate, showing you the heavy and light chains. And then the antigens bind via induced fit. So what does that mean? Yeah, you're going to get a conformational change upon binding, which helps to stabilize that, right? So antigen binding causes significant structural changes to the antibody. Antibody specificity is an important property in antibody reagents. So a lot of you guys either program you guys are going to be in, whether industrial biotech or clinical chemistry, reagent antibodies are common. you're going to use them. Western blots, ELISAs, enzyme, all kinds of different enzyme immunoassays use reagent antibodies. And this is typically, so this is an example of an ELISA. So you're going to coat a solid surface with either antigens or antibodies. This is basically a direct ELISA or a Western blot. You could use this also. You're going to block unoccupied sites. Anybody know what we use for a Western blot to block? No. Yeah, milk proteins. So, and then for most ELISAs, we're using BSA for the blocking protein. And those are there to block the unoccupied sites of your antigens. And then incubate with the primary antibody that's going to be specific against the antigen you're interested in detecting. Then there's going to be a secondary antibody that has an enzyme or a fluorophore label on it that will recognize that's specific to the Fc fragment of the species that the primary antibody was produced in. So a lot of primaries are produced in mice, so you'll have a lot of goat anti-mice. A lot of primaries also, polyclonal antibodies are produced in rabbits, although there's companies that make monoclonal rabbit antibodies these days. Then you're going to add a substrate that will react with the enzyme that's covalently attached to the secondary antibody, and then that substrate will produce a product that'll either, one of the products that could be produced, byproducts, is light, so chemoluminescent reactions, fluorescent reactions, or a chromophore reaction where you have a colored precipitate or some compound that absorbs light at a particular wavelength. And that's basically, this would be basically a direct ELISA. There's sandwich ELISAs where you actually coat with an antibody rather than the antigen, and then block again, and then use another antibody to kind of sandwich the antigen you're trying to measure in between the two. So if you're doing a sandwich ELISA, it's important that the antibodies recognize different epitopes, unless you're measuring some sort of structure that has multiples of the same epitope, like a virus, right? So virus structure, they'll have the same protein over and over and over again on that coat. So, okay. Protein interactions modulated by chemical energy. So ATP can cause a conformational change in proteins generally required for their function also, and especially in motor proteins. It'll control the movement of cells and organelles within the cells, and it'll allow for a spatial and temporal regulation of interactions. And the one motor protein we're going to talk about is muscle structure, or muscles, so actin, myosin. So muscle fibers are large, single elongated, multinucleated cells, and each fiber contains about 1,000 myofibers. So you have all these myofibers, you have about 1,000 of them, and they contain like a Z disc and an M line, and the sarcoplasmic reticulum. So, and the sarcoplasmic reticulum is important. It stores what? Yes, calcium. So normally calcium concentrations are held at really low concentrations intracellularly, but we have this sarcoplasmic reticulum acts as a store for calcium. So, and then you have the myofibrils that contain thick filaments of myosin, and they have these about 150 nanometer tail of like a helix upon a helix with two large chains on the amino terminus, and then those are bound together. They form all these, bound together like this as a full myosin fiber. And these myosin's going to interact with actin. Myofibrils contain thin filaments of actin. And F-actin, did anybody know what the F stands for? Or filamentous? Filamentous actin, because it's in a filament. G stands for, what do you think? What kind of protein is actin? Huh? Globular protein. So globular actin. So monomers of actin are globular, and then change the concentration of calcium inside the cell, and it'll form filamentous actin spontaneous. So that's how you can get a lot of rearranging of intracellular structures. But anyways, in muscle, most of it's going to be the F-actin, and this is what interacts with the myosin. Myosin head, there's binding sites on the actin filament that the myosin head will interact with. So the actin's the thin filament, myosin makes the thick filament. And structurally, they look something like this, right? So you have an I band and an A band, your Z disc, M line and Z disc. Oh, this isn't working. So it should be contracting here, but it's not. But basically, what's this model? Somebody told me about it the other day. Yeah, sliding filament model where the filaments slide along each other. So nothing's actually getting shorter or longer. It's just the filaments slide along past each other and then will come back. So, and how does that work? Does anybody know? Can you talk me through the muscle contraction cycle?
Like a zip line.
A zip line?
Yeah. Zip lock where you slide it together and close those and you open up.
Yeah, but you know how actually, why is it getting slid together? That's what biochem, I guess, is for. But there's basically 4 cycles, right? So ATP binds to the myosin, and this causes a conformational change in the myosin head, which causes it to disassociate from actin. When ATP is bound to myosin, it has a low affinity for actin. ATP gets hydrolyzed by that myosin head. You get a conformational change of the myosin, and then the myosin is going to reconnect with the actin because instead of having ATP bound after it's hydrolyzed, you're going to have ADP bound. The myosin is going to reconnect to the actin filament at a different location and then release phosphate. Release of the phosphate causes a power stroke where the myosin returns to its initial state, shifting from the actin filament relative to the myosin tail. So it kind of shifts the head shape. And then you get release of ADP. And then ATP can come in and bind again, causing it to have a low affinity for the actin. Nothing is working. Sorry. This is a nice little, what do you call it? Yeah, another GIF. My first GIF worked, but none of these work. So this is a nice little GIF that shows you through all the steps. But I'll post maybe a link to a working one on Canvas. But basically, this talks you through the steps. So one thing that seems to be left out of this presentation is the role of tropomyosin, right? Tropomyosin and troponin. So what's going on with tropomyosin and troponin? So,
Calcium binding site.
yes, there's a calcium binding site on the troponin, right? Tropomyosin just kind of holds the troponin to the actin, kind of decorates that actin chain. Calcium will bind to the troponin, causing it to have a conformational change and a low affinity for actin, and it shifts up, exposing the myosin binding sites on the actin for the myosin head to bind to. And as long as there's ATP around, it'll go through its cycles of, with ATP bound, it'll pop off. Hydrolyzed ATP, it'll go back on, release phosphate, and then it'll shift, work its way down that actin. So what releases the calcium? Yeah, acetylcholine will release calcium. But basically you get an action potential, right? Which causes voltage-gated calcium channels on the sarcoplasmic reticulum to release calcium into the cell, basically. Yes?
So the voltage-gated channels open, all the troponin, calcium binding happening, and then it goes through the 4 sites?
Yeah, so the voltage-gated calcium channels will open, releasing calcium. Calcium concentration increases, which will cause binding to the tropomyosin, the tropin, causing it to have a conformational change, exposing the myosin binding sites on the actin. So if there's ATP around, then the actin can do its thing and walk down the actin helix. The myosin can do its thing and walk down the actin helix, right?
Right. So this cycle happens at each little section of the actin helix, or just all at once everywhere?
Yeah, it's going to be going all at once, kind of everywhere.
Okay.
So long as there's calcium and ATP around. And magnesium, too. Magnesium is going to be a cofactor for the ATPase on the myosin head. So availability of myosin binding sites. Oh, so I skipped ahead, I guess. Because here we're talking about the tropomyosin and the troponin C. So availability of the myosin binding sites on the actin is regulated by troponin and tropomyosin. Nerve impulse or action potential triggers the release of calcium, causing a conformational change to the tropomyosin. And that's what I just talked about. And that's it for basically chapter 5. And then we're on to enzymes. So key topics, we're going to talk about the physiological significance of enzymes, origin of the catalytic power, chemical mechanisms of catalysis, we'll go through some of those, and mechanisms of chromatrypsin and lysozyme, and then description of enzyme kinetics and inhibition. So what are enzymes? They're catalysts, right? So what do catalysts do? Speed up the reactions without being consumed, and they don't change the equilibrium and so forth. So most enzymes are globular proteins. However, some RNA also catalyze reactions, and RNA catalyzed reactions we see in ribozymes and ribosomal RNA. So studies of enzymatic processes is the oldest field in biochemistry, dating back to the 1700s, and then enzymes has dominated biochemistry in the past and continues to do so. And these are some of the guys from history of enzymology. So I've heard of a Buchner funnel, right? So why biocatalysts over inorganic catalysts, right? So here we have a molecule. It's got an ester here, right? And it can break down by a number of different mechanisms naturally, right? But what enzymes are going to do is cause this chorismate mutase. It's going to cause it to go in one particular direction, because all of these are going to go naturally, but they're really, really slow rates. If you add something that's going to speed up the rate, it's going to go that desired direction. So you get greater reaction specificity. And why do you get specificity for?
You're able to determine the reaction rate, the amount of energy that is the reaction.
Well, not just that, but it's the protein structure, physical structure of the protein, the binding site is very specific for a particular substrate, right? So that's your specificity. Mild reaction conditions, so that's conducive inside conditions of cells, right? So we can burn cellulose and make carbon dioxide and water, but what do we get from that? Huh? Yeah, but how do we get that energy?
Oh, through a really long.
No, well, not us.
Oh.
But if we just burn it.
Oh, I thought you meant our body only.
No, yeah, our body does it, yeah, and we go through, like our body will burn sugar through each little step, we harvest a little bit of energy. But we could burn it all at once, and all that will just go to heat, right? And that's not conducive for, metabolism in our cells. So we capture a little piece of energy each step along the way. Also, for each step, let's say for glycolysis, again, doing organic chemistry, what kind of, what am I trying to say? Efficiency of each reaction, how was that during your organic synthesis? Huh? You'd be really lucky if you had 90%. Yeah. Really lucky, right? So glycolysis, how many steps is glycolysis? It's something like that. I can't remember right off the bat. But still, let's say like 10 steps of a reaction. If each step is only 90% efficient, by the end, the overall would be what? It'd be, so 10, so 0.9, and then you have 9. It'd get like maybe 10% or so. It'll be a logarithmic fashion. But anyways, enzymes have high efficiencies of their reactions. So high reaction rates, so that's useful in a biological time frame also. And then enzymes also have the capability for regulation, so there can be controlled by covalent modifications or other substrate allosteric modifications to alter which pathways are going to be producing substances and which ones won't, depending on the need of the cell at a particular time. So enzyme substrate selectivity. So what is this? Do you know? So this particular amino acid. Yeah, phenylalanine, right? So so phenylalanine hydroxylase. So here's some terminology. Hydroxylase. We're adding a hydroxyl group to it. So what is this then? Once we've added a hydroxyl group to phenyl, huh?
It's tyrosine.
Tyrosine. So, okay. Do you know what this one is?
It's the exact same thing.
Almost.
No,
It's the stereoisomer, right? So this, you don't have any binding of the phenylalanine hydroxylase, right? Because it's stereospecific. And in this, you get binding but no reaction, so this would be a good enzyme inhibitor, right? So very specific and very selective for what it binds to. So. Reaction conditions compatible with life, right? 37 degrees and a pH of about 7. So what happens normally when you increase the temperature of a reaction, what happens? Speeds up the reaction. Why does it do that? I think we talked about it on the first day. Yeah, so if you have the reaction coordinate, you're looking at it for a particular reaction. This is at one temperature. If we just heat the temperature or heat the reactants, they have an overall free energy here. And actually it'll probably be, products will have the same increase in temperature. But your differences here would be smaller for the average. What's this called? Free energy of activation, right? So that's how you speed up the reaction. But what happens, why can't you'll speed up the reaction as you increase the temperature with an enzyme? What happens as you increase the temperature?
The enzymes are in nature.
Yeah, the enzyme unfolds and you'll lose that activity. Same thing for pH. So a lot of enzymes have a specific pH that they work best at. For example, pepsin in this graph has its highest activity of about a pH 1. Where do we find pepsin? In our stomach, right. And then other ones, a lot of them have their highest activity around pH 7, which for the most part is conducive for compatible conditions with life. 6 classes of enzymes defined by the reactions that are catalyzed, and this is the overall reaction coordinate that we see with enzymes, right? So for an uncatalyzed reaction, we have the transition state, so that's the energy of activation we have to overcome to get down to the products. Enzyme catalyzed reaction, there's a binding event where you form an enzyme-substrate complex, and then that's going to go on and produce a product. You have an enzyme product complex, and then release of that product, you get free enzyme and then the free product. So, and we can look at this mathematically. Have you guys ever heard of the Michaelis-Menten equation? No? I always fail at this. Right? So if we want an equation that just tells us what the velocity should be for a particular substrate concentration, we know the reaction is basic. For a basic reaction, we have an enzyme, free enzyme, plus a free substrate. That'll form a reversible reaction governed by a rate constant K1 or K minus 1. These are little k's. That's going to form this ES. And then that pretty much, for most reactions, is just going to go form a product. Okay, and let's just take the, basically, we know enzymes, or the reactions they catalyze are reversible but for most of what we're going to be doing, we can look at them as irreversible. And we want to come up with an equation that tells us basically what the rates would be for a particular substrate concentration, not changing the enzyme concentration. So we know, let's look at this. The rate going this way, so K formation, or the rate of formation equals K1 times the concentration of enzyme, free enzyme, times the concentration of substrate. And then we also know the rate of how this disappears is going to be equal to K minus 1 times the concentration of ES plus K2 times the concentration of ES, right? So at steady state, these two rates are going to be equal, right? Does that make sense? So if we set these equal to each other, we're gonna, because this is typically the slow step going here. We're gonna put all the constants on one side and then all the concentrations on another side, right? So we get K1, and this will separate ES. Divided by K minus 1 plus K2 equals ES divided by okay, and then this is just all constants, right? So we can just make one big constant for that. And then one other thing is the amount of free enzyme is going to be equal to what? Or we have total enzyme, right? It's going to be equal to the concentration of your free plus the concentration of your ES. Does that make sense? So
how do you calculate the concentration of K to the catalyst? How do you calculate the concentration of K to the catalyst?
The which K? This?
Yeah, the K catalyst.
That's going to be basically it's related to the Km, or basically is the Km. Oh, the K cat here.
Yeah, the K cat.
So the K cat is basically it depends on the slow step.
Right. So it's
And the K cat times your total enzyme concentration is basically your V max.
okay.
The fastest, the velocity that you can have. Does that make sense?
It does. So the velocity of the reaction, or the rate at which the reaction goes on, depends on the catalyst of the slowest reaction.
Well, it depends on which enzyme it is. Basically, it's going to depend on how much enzyme is there. But typically in a system, when you're making a graph, so the velocity of product formation is going to be, you're going to relate that to the enzyme or the substrate concentration, and you're going to see a graph like this. So we saw something like this last time, right? It's your basically your binding effects, right? But this is going to be your V max, which is your theoretical. It's a theoretical velocity that's the maximum that you could see with that amount of enzyme. No matter how much substrate you add, you'll never reach your V max or pass your V max. It's the fastest the product can achieve. So your K cat is basically this, basically the rate, the slowest K. It could be K2. You can actually draw this with a K minus 2. And we can go a step further and make this EP. And then that has a 3 and a minus 3 with free enzyme. But to do the Michaelis-Menten, we're not going to involve that, because for the most part, this K2 is the slowest reaction. So K2 normally is going to be equal to the K cat. Does that make sense? And you can actually calculate that by determining what your V max is, right?
Yeah. And once you calculate the V max, it's just...
Yeah. So, and the V max, I mean, you're not going to ever pass it. So, where was I? We were trying to calculate this equation, right? So we want to get rid of all the ES, because we just want enzyme concentration and substrate concentration. So enzyme total subtracted by the total enzyme, or free enzyme, is going to equal to ES. Okay, so we can substitute that into here. Well, I don't have enough board to do a good one of these. Wait, I meant total, right? Not total, free enzyme. I don't know what I'm doing. So this is basically the Km. And then we want, where was I going with this? I did this earlier today, but then got distracted again. Here, I got it all in electronic form, right? Here. Somewhere. Right here. So we got total enzyme, which equals your free enzyme, plus your enzyme-substrate complex. Velocity forward is going to equal the K1, or this. Rate constant times E total times subtracted by the ES, which is what? Basically your total free enzyme. So that's where I messed up. So K forward is going to be K1 times which is also equal to K1 Okay. So you see where that comes from then, sort of? No? So the velocity of dissociation, K minus 1 equals K minus 1 times enzyme-substrate complex plus K2 times enzyme-substrate complex. And at steady state, those two are equal. Therefore, we get this. And then we solve, put all the K's on one side and the E's, S's and E's on the other, and we get K1 divided by K minus 1 plus K2 equals the enzyme substrate complex divided by total enzyme subtracted by ES times the substrate concentration. Okay? And that basically gives you, all of these constants can combine basically to tell you what the Km is, which is basically analogous to a KD. Anybody remember what the KD is? Dissociation constant. So do you get stronger binding with large dissociation constants or small dissociation constants?
Stronger binding with small dissociation.
Yeah, small dissociation constants. Okay? So then we can rearrange that to get this, because we're trying to solve for the ES. So basically, so the velocity of production is going to be K2 multiplied by ES, and that's going to be equal to, well, first we solve for ES. Do you guys see how we solve? I don't show it there. So we're going to have to rearrange this to get ES out of it. So we'll put ES, Km on the bottom here. And we're cross multiplying basically. So we have enzyme total minus ES times the substrate. And if we multiply through there, we get ET times the substrate concentration minus ES concentration times the substrate concentration. And then we'll just add ES/S to both sides, right? Wait, no, we want to whole this out of here. What am I doing? No, we'll just do that. So we get okay, now we can pull ES out of that. Where do I do this? And we get ES times 1 over Km plus the substrate concentration, and that's equal to, where are we? ETS. And now we divide by this, and we get enzyme substrate concentration equals ETS divided by 1 over the Km plus the substrate concentration. Okay? Which, where did I get 1 over Km? What, do you know? You raised your hand.
No, I'm just like, you've been at it for so long. Like, I've now lost the point.
Okay.
Yeah.
Well, basically, we're trying to solve for ES. So we want an equation that tells us the velocity forward, which is going to be K2 times the ES. And when ES equals this, it's just K2 times this. But I don't understand what I did wrong. Somewhere where I transferred it. I think I should have just left Km over here.
I think it may have been the ES multiplied by 1 divided by Km plus S.
Yeah.
And then it's ET.
So I have ES divided by Km plus ES. Let's multiply Km here, both sides. You have E Km ETS equals ES plus ES S Km. Pull ES out of that, and we have times 1 plus SKM. Let's multiply for that, and we get Km ETS divided by 1 plus SKM. And we got to get rid of some of these KMs, so if we multiply by, if we just pull Km out of this, again I get 1 over Km, so I must have made a mistake somewhere, nobody knows where. Well, I've done this 100 times too, I don't know why I messed up somewhere, but we got to keep going. So you solve for ES, and then the velocity of product formation is K2 times the ES, which you end up with that equation. And what happens when the substrate concentration is much, much, much greater than the enzyme total? ES equals basically enzyme total, and you get velocity forward equals V max. So basically V max is going to be K2 times enzyme total, or what your book is saying This is the same thing as Emax2. It's the Kcat times the enzyme total. So enzyme-substrate complex, this basically shows you an image. There's key active site amino acid residues that are going to be able to specifically bind to those substrates, and they stabilize the transition state, basically. And that's how they do their catalysis. Enzymes do not affect equilibrium. Slow reactions face significant activation barriers, which is the change in free energy of activation that must be surmounted during the reactions. Enzymes increase the reaction rate by decreasing the activation energy. And do you guys remember this equation from general chem? Equilibrium, basically your energy of activation, dependent on K. Or that's how you can change that. So, anyways. And this just shows you a normal reaction coordinate. I don't know how many times I've had these up here. With a decreased delta G, so we already showed you this, you have an enzyme-substrate complex, enzyme-catalyzed free energy of activation, and it's much smaller than your uncatalyzed free energy of activation. And then your table with a number of increased rates. Not increased rates, but rate enhancement. So carbonic anhydrase, you're 10 to the 7 times faster than you would be, if that reaction would be occurring naturally. Same thing with urease, the enzyme that breaks down urea. 10 to the 14 times faster than it breaks down naturally. So how does this happen? So for example, one of the methods, if you have an uncatalyzed bimolecular reaction, what has to happen? Do you remember what a bimolecular reaction is?
Two molecules react.
Yeah, so two molecules coming together at the exact right orientation and velocity where they can get the reaction to occur, right? So enzymes get past this. This is entropically unfavored. You got to put a lot of energy into it, so there's two molecules will have enough kinetic energy that'll collide at just the right angle. Two free reactants, single restricted transition state conversion is entropically unfavorable. So an enzyme is going to help bring these two together. It in effect increases the concentration and assures that they're going to be in the right orientation based on the specificity of the binding site of that enzyme. Does that make sense? So uncatalyzed unimolecular reactions, you have a flexible reactant, rigid transition state conversion, and that's entropically unfavorable for flexible reactants, right? Because you got to get over that transition state to get to to have that reaction proceed. And what an enzyme does for a catalyzed reaction is it stabilizes that transition state. So catalyzed reactions, enzymes use the binding energy of the substrate to organize the reactants to a fairly rigid ES complex. And they're going to have that, bring the local concentration of both higher and give them the right orientation and put them, where they can react. Entropy cost is paid during the binding. You get a rigid reactant complex. Transition state conversion is then entropically okay. And this is some support for that proximity model. That's what I was talking about. Enzymes bring them close together. If you react, you have acetic acid versus some kind of acetic acid ester to get released to form an acetic acid anhydride. It's going to be a very slow process with just these around. But if you have a molecule where you have an intramolecular structure such as this is much faster because these two are close together. They're part of one molecule. So this molecule will break down, release this alcohol, or oxide rather, much faster than you would if you just have acetic acid and some acetic acid ester. same thing for this. So this totally has them in the right orientation. You got even less rotation around this. They're kind of fixed due to this cyclic structure. And that converts to this, 1,000 times faster. So, does that make sense? So that's your proximity, support for the proximity model. So how do they lower the free energy of activation? So Linus Pauling hypothesized that enzyme active sites, instead of being complementary to the substrate, they're actually complementary to the transition state. So they, in effect, bind the transition state better than they do the substrate by lowering that free energy, the energy of that transition state. What's the definition of a transition state? Anybody give me any definitions?
Like carboxylic acid.
It's not necessarily carboxylic acid, but it's like
Step between two.
the step between two reactants and products, right? So transition states are typically inherently unstable. You can't capture them as a transition state and then purify that transition state because they're so unstable. So if an enzyme preferentially binds that transition state, it's kind of the induced fit model, right? You'll kind of convert the substrate into the transition state, but because it's kind of forcing it to be in that transition state, it's inherently unstable. It's just going to go forward and proceed with whatever reaction it's catalyzing, right? Does that make sense? So they bind transition states better than substrates, and stronger additional interactions with the transition state as compared to the ground state lower the activation barrier, and it's largely a free energy, free enthalpy change of transition state. This is Linus Pauling. And these are the figures of this book that I really like, because this is kind of puts it like a visualization of how enzymes would work, right? So say we have this metal stick, and we want to produce two broken sticks, basically. The transition state between those two is going to be a bent stick, right? So if you have an enzyme that's complementary to the substrate, it'll just bind that straight stick and just sit there, because the free energy of the enzyme-substrate complex is much lower even than the product. You won't get any, you've actually increased the free energy of activation. If that enzyme-substrate complex are so happy energetically being together, there's no way you'll get that to go through the transition state and form a product. Does that make sense? Whereas if you have an enzyme that inherently binds the bent stick rather than the straight stick, it'll bind the straight stick but then try to force it to form a bent stick. But the bent stick's going to be, when you bend it so much, it's going to break. And that's how you get the enzyme to produce the products. And you get a reaction coordinate looks like this. It binds enzyme-substrate complex is a little higher in energy than the substrate alone. But then that lowers that transition state energy because you have this big magnet pulling that metal stick to bend. But then, of course, what would make that magnet want to unstick from the broken metal sticks? But still, I like this model. So catalytic mechanisms include acid-base catalysis, covalent catalysis, metal ion catalysis, and electrostatic catalysis. So acid-base catalysis, of course, is going to use like the transfer of protons between either water, product, or R groups or amino acids in the protein itself. Covalent catalysis involves a covalent bond between something, one of the reactants, and the enzyme itself. Metal ion catalysis, typically there are a lot of redox reactions, so it's going to require metal ions. A lot of them are going to be magnesium, things like that. Electrostatically preferentially interacts with the transition, or the ES, transition state. So here's an example of acid-base catalysis. Got some acid. It's going to react with the carbonyl group. And then you have a transition state with this. It's going to be unstable. So a proton transfer to or from water It's faster than the rate of breakdown between the intermediates. So you have this, the proton is going to go to water this way, or a base. A lot of times when you see the enzymes, the mechanisms written, you have bases, just a B stands for a base on the enzyme itself that can accept hydrogens or donate hydrogens. And then you get the reaction to continue, and you get the products like this down here. And we're going to specifically look at serine proteases in a second. Well, it just moves around. So amino acids and general acid-base catalysis, of course, glutamic acid and aspartic acid, they have carboxylic acid group as a proton donor, and the carboxylic, the conjugate base can act as a proton acceptor. Lysine and arginine have the amine groups that can accept and donate a proton. Cysteine has a thiol group. Histidine has this group here that can accept and donate a proton. Serine, normally you don't think as alcohols as acidic, right?
No.
Yeah. In serine proteases, serine itself gives up this proton. Tyrosine, phenol is an acidic compound, right? So it's and the tyrosine is actually more acidic than phenol itself. So it can give up that proton. Remember, the negative charge gets stabilized by the aromaticity. So covalent catalysis, you have a transient covalent bond between the enzyme and the substrate. We'll see with the serine proteases, it's part acid-base catalysis and covalent catalysis. Changes the reaction pathway without an uncatalyzed reaction. In this example, we have A that's bound to B, gets hydrolyzed, and you have A plus B. Catalyzed, A, B reacts with some X on an enzyme. You have an AX intermediate plus B goes away, then water comes in and replaces A from the reacting part of the enzyme. So it requires a nucleophile on the enzyme, can be a reactive serine, thiolate, amine, or carboxylate. So, like I said, metal ions involve metal bound, and it's going to interact with the substrate to facilitate binding. Stabilizes negative charge, mostly, those metal ions. So magnesium, 2 positive charge is going to be able to stabilize negative charge, and it'll participate in oxidation reactions. So chromotripsin uses most of the enzymatic mechanisms. Chromotripsin, anybody know what chromotripsin is? So it's a protease that specifically will cut peptide bonds after aromatic amino acids. So there's a number of parts. There's this catalytic triad that includes an aspartic acid, a histidine, and a serine. There's a binding pocket, hydrophobic binding pocket that will bind the aromatic R group, and there's an oxyanion hole that stabilizes an oxide that's produced as an intermediate. So, and this is the active site of troine. This is your catalytic triad. You have your aspartic acid, a histidine, and a serine. So the aspartic acid is going to draw protons away from the histidine, causing the active site of the histidine to act as a stronger base, which will draw away the proton from the serine, allowing the serine to become a better nucleophile, and that'll attack a carbonyl and a peptide bond. So this is the hydrophobic pocket, and then you have a glycine up here where the peptide bond itself stabilizes an oxyanion that forms. So here we have the aspartic acid drawing a proton from a histidine, which then draws the proton from the serine, which is going to allow this serine to attack, as we see here, these electrons from the histidine, grab the proton from the serine, allowing the electrons from the serine to attack the carbonyl and the peptide bond. And you form this oxide, so you have the NH from the serine itself and the NH from the peptide bond and a glycine. That helps stabilize the negative charge that then forms there. And you have this tetrahedral intermediate that forms. It's a tetrahedral oxyion that forms, and it'll collapse. The electrons go back down to the forming a carbonyl, and you're still covalently bound on this carbonyl to the serine, where the electrons in the peptide bond will then pick up the hydrogen to form the base, and you have a new amino terminus on a peptide that was just cleaved by the chromatrypsin, and then a covalent intermediate that will have a new C terminus on the N-terminal side of the original protein. So that's how you get substrate cleavage. Your first product comes off with a new amino terminus. The next step is water has to come in and hydrolyze the bond between the serine and the rest of the peptide. So it's acyl enzyme intermediate. Water comes in, attacks the carbonyl, performs nucleophilic attack on the carbonyl. And again, you have a tetrahedral intermediate. It collapses. Wait. Yeah, water attacks. Oh, I'm jumping ahead. The arrow's going this way. Water attacks. You form a new tetrahedral intermediate. It's going to collapse. When it collapses, you know, these electrons force these electrons to pick up another proton, and the serine is restored to serine. So you have deacylation. You have the serine there again as an alcohol, and then you have the free peptide with a new C terminus. Does that make sense? And then that will unbind, and you have the regenerated active site also. So that's your serine protease's mechanism of chromatrypsin. So peptidoglycan and lysozyme. Peptidoglycan is a polysaccharide found in bacterial cell walls. Cleavage of the cell wall leads to the lysis of bacteria. Lysozyme is an antibacterial enzyme. Do you know where we find lysozyme?
In the tears?
Yes. So lysozyme is a protein we produce. It's found in our tears, and it's to inhibit bacterial growth in our, eyes. So peptidoglycan and lysine. Peptidoglycan is a polysaccharide for the most part. And lysozyme just hydrolyzes the bond in between the two sugar moieties. So X-ray structures of lysozyme with bound substrate analogs show that the C1 carbon is located between glutamic acid 35 and aspartic acid 52 residues, right here and here. And you have a covalent intermediate that's bound to the active site in that image. So aspartate 52 then acts as the nucleophile to attack the anomeric carbon. You guys don't know what an anomeric carbon is yet, because we haven't done the sugar chapter. So we'll talk about that later. The anomeric carbon is the carbon that forms glycosidic bonds. So it acts as a nucleophile to attack the anomeric carbon in the first SN2 step. Glutamic 35 then acts as a general acid. It's going to protonate, leaving the leaving group in the transition state, and water hydrolyzes the covalent glycosylyl enzyme intermediate. Glutamic 35 then acts as a general base to deprotonate the water in the second step. And this is the overall reaction here, right? They're the overall first steps. So you have glutamic acid, 35. It's going to act as a general acid, donating this hydrogen So aspartate is going to attack the anomeric carbon here, allowing these electrons to then grab that proton, and you have the first product leave and a covalent intermediate. The next step water comes in. The glutamate residue 35 picks off a proton from that, allowing hydroxide to attack that carbonyl, or that anomeric carbon, which then the electrons go back to the oxygen on the aspartate. And you have your second product leave, and then the glutamic 35 is protonated again, and the aspartate is deprotonated like before. So that's the overall reaction for that. What's the difference between these two? So, what is enzyme kinetics? Kinetics is the study of the rate at which compounds react. The rate of enzymatic reactions is affected by the enzyme-substrate effectors and the temperature. We're almost out of time. So we'll do the enzyme kinetics next time, I guess. That's where I was going with this anyways.